Hi, here we have another lecture, this time on photosynthetic light reactions and photoinhibition. Before we start talking about photosynthesis and specifically about the light-absorbing reactions, I want to take you back to some chemistry that you probably heard about in your introductory chemistry class, and that is the redox potential. So the redox potential is important for this lecture because many of the steps that are involved in light absorption and creating chemical energy from light energy are redox reactions. And so I want to remind you what a redox reaction really is. So in this example here, you have a double container. This is a glass container, contains iron sulfate and an iron electrode in here. And then inside is a porous container, and that contains copper sulfate inside, and it contains a copper electrode that is inserted into the solution here. And in between is a wire and a voltmeter. So you can measure the voltage of any electricity that flows between these two electrodes. So if you set up this experiment, you will be able to measure that electrons flow from iron to copper. Now, if you were to change this setup and you were to use the copper, but instead of iron, use silver, like so, where you have silver and a silver electrode and silver sulfate on the outside, the electron flow is reversed. Now, electrons are moving from copper to silver. So you might have heard about precious metals and how electrons are moving between metals. That is the reason for why electrons are moving towards copper in one case and away from copper in the other case, because it's always a question of what the other substance is that the reaction occurs with. So the potential, so the ability of the electrons to flow, that's the redox potential, and that depends on the chemical properties of the compound or the elements or the substance that you are measuring. And so silver compared to copper will allow electrons to flow towards silver, while if you compare it to iron, the electrons will flow in the other direction. So for our intents and purposes in biology and the discussion of light reactions and photosynthesis, there are two general rules I'd like you to keep in mind. So electrons are transferred from one compound to another depending on their chemical properties. So that is the redox potential of these substances between which the electrons flow. And the second is sort of a rule of thumb that tells you if you add a hydrogen to an organic substance, then that in all practical terms really is a reduction. So if you're thinking about is it an oxidation or is it a reduction, you can use oxidation numbers in order to figure it out. But in most cases in biochemistry, reduction in practical terms means the addition of a hydrogen. So you add a hydrogen, that is a reduction reaction. All right, so now let's look at some physics. So when you have chlorophyll, and you're looking at the absorption spectrum, which we will a little more detailed in a second, you will see that chlorophyll can absorb light particularly well in the blue and in the red part of the visible spectrum. So the wavelengths are here on the y-axis, 400 to 900. So this is the blue spectrum, here's the red spectrum. And in that part of the visible spectrum, that is where chlorophyll absorbs electrons. Now over on this side here, on the left-hand side, you see energy content on the y-axis, and you see something that's called the ground state or the excited state. So when light strikes chlorophyll, light is absorbed, and the electrons can move to a higher state. So they move a little bit further away from the nucleus, and that requires some energy, and that's the energy that they pick up from the photons, from the light that strikes the chlorophyll. So light strikes the chlorophyll, electrons are being propelled to higher excited state. Now, what happens to the electrons in this higher excited state? There are a number of things that can occur. One is that the electrons move back to a lower state, and as they do so, they lose energy, and they do that via heat loss. So light energy is transformed into heat energy. There's also the possibility with some substances that the energy of these electrons, as they fall back to the ground state, emit this energy not in the form of heat, but in the form of light. So if they absorb, if they lose the energy in terms of light, then they will fluoresce. So fluorescence is the energy, the type of energy into which the energy originally that came from the sun and that was moved into an upper high excited state is then converted as the electrons fall back to the ground state. So heat and light. But there's a third state, and that third state is really the one that we're interested in this specific discussion. So electrons can move to a different substance, a different state, a different part of an overall larger compound. And as they do so, rather than just jumping up and down between these two states, they can sort of like a slinky down its stairs. They can go from a higher to a lower level, but they don't have to go all the way in one step. And so each time that they move from one level to the next level, to the next level, to the next level, they lose a little bit of energy, but they can give that energy to the next substance, changing the substance in a redox reaction, and using that to convert certain substances so that they can do certain types of work. And you'll see in a second what I mean with that. But the point here is that chemical energy can be made in these multiple steps in these reactions where the electrons don't just lose their energy as heat or as light. So let's sum this up, what I just said. Forms of energy loss after excitation, either as fluorescence or heat or transfer to another compound. And you will see in a second that the ones that we're interested in are the chlorophyll compounds, and then eventually to the electron transport chain in which the energy in these electrons is converted to photochemistry, to substances, eventually to sugar. Okay, I said we were going to look at the photosynthetic pigments, at chlorophyll a little bit more in detail. So here is an absorption spectrum. Last week you looked at an absorption spectrum for phytochromes. Here's an absorption spectrum for photosynthetic pigments. And so you see on the x-axis the wavelengths within the visible spectrum from blue via green to red, and then the amount of light, relative amount of light absorbed on the y-axis. Chlorophyll A and B have relatively similar absorption maxima in the orange and red, and slightly different absorption maxima in the blue. So chlorophyll A absorbs a little lower wavelengths, chlorophyll B absorbs a little higher wavelengths, but you also see that there's overlap, right? So you see these shoulders that are overlapping. So this entire area, the blue-green light, is absorbed quite well by chlorophyll. And then you also see the carotenoids, which can help chlorophyll by absorbing light in parts of the spectrum that chlorophyll is not so good at absorbing it. As you can see that this part of the spectrum is covered by carotenoids, which can then transfer the electrons and the energy that comes with them to chlorophyll in subsequent reactions. So chlorophyll absorbs the red and blue part of the spectrum, carotenoids absorb electrons and pass them to chlorophyll, and then chlorophyll can use them from there on. And just as a reminder, what we talked about last week, an absorption spectrum describes the ability of a compound to absorb light at a given wavelength. Here's the given wavelength. Here's the ability of this particular compound, in this case chlorophyll, to absorb that kind of light. All right, so last week we also talked about another type of spectrum. It wasn't the absorption spectrum, but the action spectrum. So an action spectrum tells you something about the action of certain wavelengths in a given system. And so there's a classic experiment that was done by Engelmann in 1883. And what he did is he took light, just a white light source, and he passed it through a prism. And the prism breaks it up into all the different wavelengths. So the blue, the purple, and the blue, and the green, the yellow, orange, and red. And then he took a green alga, so a photosynthetic organism, and this alga is a sort of a long, spirally, stringy kind of alga, and he strung it along this the area into which the light had split. So if you look at the prism, you look at the light that's coming out sort of in a cone. He strung these alga, Spirogyra alga, along the different light colors. And since these alga, we now know that, of course, produce oxygen during photosynthesis, he wanted to know what wavelengths were particularly effective. And so what he did is he inoculated this alga with aerotactic bacteria. So these are bacteria that need oxygen. They like oxygen. They move towards oxygen. So they can grow there. They move there. They will accumulate in areas where there's oxygen available. And so you see that these dots here are supposed to be numbers of or individual bacterial cells. And you can see that they accumulated in the blue area of the spectrum and in the red area of the spectrum, in the areas where the alga was strung along those colors that had been passed through the prism and split up into the different colors. So along the blue colors, the blue wavelengths, there were bacteria that were picking up oxygen, and along the red one. And so he could say from this experiment that the action spectrum, so where do we have action, where do we see oxygen evolution occurring, occurs here in the blue and in the red. So chlorophyll action with respect to oxygen production, or photosynthesis as we now know, occurs in the blue and the red parts of the spectrum. And just as a reminder, the definition of an action spectrum compared to an absorption spectrum describes the effectiveness of a particular wavelength on a specific response. All right. So the pigments that absorb the light are chlorophyll. There are two types, chlorophyll A and chlorophyll B. They contain a porphyrin ring, and I have a figure of that in a second. They have this long carbohydrate tail, and they're complexed with magnesium. And so here you see an example of where magnesium is required for the plant to have as part of their nutrient intake. And then we have the carotenoids, which are found in higher plants. And in algae, we find another accessory pigment, just like carotenoids in higher plants. In algae, this accessory pigment is phycocyanin or phycoerythrin. All right, let's take a look at a chloroplast. So just so that we have all the terminology straight, here's our plant cell. And these green blobs here are the chloroplasts. And then if you look at a chloroplast has a double membrane on the outside, but it also has inside membranes. So it has the outer membrane and the inner membrane. And then inside the chloroplast is some sort of substance. You would call it plasma in the cell, but the plasma in the chloroplast is called the stroma. So the stroma is the liquid inside. And then there's this other membrane system inside, and that is called the thylakoid. The thylakoid membrane makes up the membrane, the outside, and then inside these thylakoid membranes is the lumen, the thylakoid lumen. And a stack of these thylakoid membranes on top of each other, that's called a granum or a granum, a stack of thylakoids. And if you take this little piece here and you blow that up and move it over here, then you can see the membrane, that's the thylakoid membrane here, and then the stroma, so that's the plasma, and then the inside, the lumen of the thylakoid. And all these little blue, purple dots here, that is ATP synthase. And as we will see in a moment, that is where the hydrogen gradient with high concentrations of hydrogens on the inside, lower concentrations on the outside, due to the splitting of water and creation of a hydrogen gradient in the electron transport chain that I will discuss in a second with you, that is where ADP is changed into ATP, and where energy essentially is created, chemical energy is created. So here's another view of the chloroplast. You see the thylakoids here, you see the membrane, and here's a figure of that of the chlorophyll. So this is the chlorophyll molecule. This is where magnesium is complexed. This part is called the porphyrin ring. 1, 2, 3, 4 parts of this ring. That's the head molecule, and then here's the hydrocarbon tail. And then this part here is the only thing that's different between chlorophyll A and chlorophyll B. So either you have a methyl group there, or you have this aldehyde. All right. So the chloroplast contains the stroma. It's pretty much the plasma of the chloroplasts. And then you have the thylakoids that contain the chlorophyll. Inside is the lumen. And the photosynthesis reactions, the light reactions occur on the thylakoid membrane. Okay, and that is what we want to look at next. So we're going to look at these photosynthetic reactions that occur on the thylakoid membrane. I assume that in BIo332, maybe even in cell biology, you've looked a little bit at the light reactions and you've seen something like this before. So let me walk you through this figure so that you can see the main parts of the light reaction and the major aspects that are important to recognize. So there are 4 major complexes. There's the photosystem 2, which ironically is the first one in this reaction, but it was the second to be discovered. That's why it's called photosystem 2. And there's a complex called cytochrome B6F in the middle of this pathway. And then there's the photosystem 1. And then we have the ATP synthase. So what happens along these membrane complexes? There are some shuttles, electron shuttles in between that allow the flow of electrons from one to the next. And remember that I said at the beginning that electron flow is really nothing other than redox reactions. So you have redox reactions where electrons are shuttled from one substance to the next substance to the next substance to the next substance to the next substance. And then eventually there's a reduction of NADP plus to NADPH. And then during that time, hydrogen is either pumped through the membrane or produced during the splitting of water. And then that sets up the electrochemical gradient, the hydrogen gradient, that is used through ATP synthase to produce ATP from ADP. Let's go through this step by step. So it all starts with the splitting of water. Oxygen is produced here. So this is why plants evolve oxygen. This is why plants produce oxygen. This is the side product. They have to get rid of it. It comes from water, right? Oxygen comes from water. And the other thing that comes from water is this hydrogen ion or proton. So keep those two in mind. They will be used later on. What's left is an electron, right? So there's a negative charge here. You have a positive charge. Here's your negative charge. So that electron is now propelled into a higher electron state. So remember at the very beginning, I showed you this physical diagram where electrons move to a higher state. This is what happens here. So electrons pick up the light, and that energy from the light propels them to a higher state. And they can now be used to reduce, so using the electron plus a hydrogen, here's a hydrogen, to reduce a substance that is called plastoquinone to plastohydroquinone. So the reduced part is called plastohydroquinone, and the oxidized state of that compound is just simply plastoquinone. And as it's doing that, as it's doing the reduction, so you add an electron or you add a hydrogen, whichever way you would like to think about reduction and oxidation, where does this hydrogen come from? It comes from the outside, from the stroma. So from the outside, not the inside of the lumen, it takes this hydrogen, and in this process, you will see, and I'll show you that in a little bit more detail in a second, that this hydrogen is taken from the outside and then later on spit out back here into the inside of the lumen while the electron travels through this system. So here's an important hydrogen pumping mechanism that uses this electron gradient, this electron redox gradient, the energy from that for this pumping of the hydrogen. All right, so we have oxygen that is evolved, we have hydrogen that is pumped, we have electrons that are reducing plastoquinone to plastohydroquinone, and they then move through the cytochrome 6 complex and then move to another substance that's called plastocyanin, which they reduce. And at that point, that substance can then reduce the next compound down the line that has a progressively lower energy value. So that's the redox potential, and ends up in this photosystem 1. And photosystem 1 can add more energy to the essentially to that electron, to that system where it's at this point. So it can absorb more light. It's another photosystem. It's another jolt of energy, propelling this electron yet to another part of the compound, another area of this large photosystem. And at that point, it goes to ferredoxin. So it reduces ferredoxin, and then ferredoxin can use hydrogen and NADP plus its energy, its electron, and reduce the NADP plus to NADPH. And so that is where the energy, where the electron in the end goes. So it flows through all of these different redox states. And then in the end, ferredoxin, that's a protein that's part of this photosystem 1 complex, uses the NADPH, adds the H, and produces NADPH. All right. Okay, so there are some parts within this. This is the linear part of the pathway that I told you about. And so there are some circular parts of the pathway. So it turns out that electrons can either flow in this linear way that I showed you, or there are some places where the electron can sort of flip back. It can circle back to an earlier state. And as it's doing that, it is allowing the system, the membrane essentially, with these compounds to pump more hydrogen, because it's basically allowing the reuse of that energy of that electron. Now, how does that work? So there's one part that's circular, where we have circular electron flow, and that's called the Q cycle. And we'll go into a little bit more detail here in a second. And there's another part where you have hydrogen flow, and you have hydrogen flow this way, later on hydrogen flow that way. Here's your Q cycle, where the electrons can go in a cyclic way in a second place, and that is called the cyclic electron flow. Sorry about these terms, I didn't make them up. They're both cyclic, but this one's called the Q cycle, and that occurs in the cytochrome complex and the plastoquinone complex. That's called the Q cycle. We'll come back to that in a second. And then if electrons flow back from photosystem 1 back to cytochrome B6F, that's called the cyclic electron flow. Okay, so that's the terminology. Now let's take a closer look at all of these parts of this light reaction. Let me just point out again, the major compounds that are produced here are oxygen through the splitting of the water, NADPH, so the reduction of NADP plus to NADPH, and ATP in the ATP synthase in the end, through the use of the electrochemical potential gradient from hydrogen that was pumped or split up from water through the ATP synthase, producing ATP in the end, right? So you should definitely remember that the three products are ATP and NADPH and oxygen. Okay, so the major proteins and the protein complexes I just mentioned that are involved in the photosynthetic light reactions are the photosystem 2, light absorption, water splitting, the plastoquinone, the cytochrome B6F, plastocyanin, photosystem 1, ferredoxin, and ATP synthase. These are the major protein complexes. All right, now we'll look at them each individually, one at a time. So let's take a look at the water oxidation step, the very first step. So the proteins that are making up that complex in which water is split, there's a particular part of that complex that's called the water splitting enzyme. So this is a major protein complex, meaning there's multiple protein parts of that. And what this is showing here is really only a part of the complex, so part of a protein. You can see that all of these areas here and then this over here are actually the protein, the amino acids from this protein that are arranged in a certain way around a cluster of manganese and oxygen. So this is the manganese oxygen cluster. This is all part of the protein. This YZ is also part of the protein. It sort of forms a little pocket where the oxygen can be held. This is also part of the protein again. So here, this Y, right? That's the tyrosine hydroxyl residue of the photosystem 2, which is the electron acceptor. So this is part, sort of around here is the protein, and then here we have the this manganese cluster that's part of the protein. So if you look closely, you see that we have two water molecules here, H2O, right? So those two water molecules are added into this little pocket. And then in this pocket, they're held there because of charges, partial charges. And you can see that the manganese is part of this, this pocket that allows the holding of the water. And of course, manganese is a micronutrient we discussed before. So you can see here why manganese is important, without which plants can't survive. So the next step is that electrons now are coming off one at a time sequentially from these two water molecules. So one electron at a time, another electron are coming off sequentially from these two water molecules that were held in that pocket. And then in the end, that oxygen that is left, here's oxygen, so that is released. You see that this part of the protein, right? This is the tyrosine part of the photosystem 2 protein. So that is now no longer bound. Here's the interaction. It's no longer interacting with the two water molecules because all the hydrogens are gone, along with the electrons, and all that is left and is now still being held are the oxygen, the O2, by the manganese. Now that oxygen is released here, and two new water molecules are added to the system. And if you add that all up, what I just told you about, you have two water molecules at the beginning, 4 hydrogen ions, protons, that are being split off sequentially, along with an electron, right? That's the electron that's propelled into the upper, into an upper level, higher level, that then is used for the redox reactions subsequently in the steps along the thylakoid membrane. So the 4 electrons and the oxygen. So in summary, water is held stably by the manganese and complex. Protons are released. Oxygen is released eventually. And then as a summary statement here, water oxidation releases oxygen without forming reactive intermediates or reactive oxygen species. And so what that means is that instead of just oxidizing the water without holding the oxygen during this process, oxygen could be released and it could lead to the formation of radicals, right? So you have an oxygen that is negatively charged by the addition of an electron. So that would be oxygen radical or a reactive oxygen species. And as we see in a little bit further later, reactive oxygen species can be very dangerous for any kind of tissue, animal or plant tissue. They're very reactive, and so they destroy stuff. So the plant is saving itself from these reactive oxygen species by holding the oxygen in this pocket with a manganese so that it cannot form reactive oxygen species and is in an orderly way released to produce oxygen that can then be transported out of the plant. All right, so that's the first step, right? So that's water oxidation along this, this enzyme complex. So the second part along this transport chain that I pointed out at the very beginning are the quinones or plastoquinones. Here's the chemical structure of such a plastoquinone. And if we start with plastoquinone and you're adding an electron, then there's a transition state where you have an electron here, and you're adding another electron here. Along with two hydrogens, you are now adding a hydrogen here and a hydrogen here. And so you go from plastoquinone to plastohydroquinone. So that is why this, this substance, this chemical, is called an electron carrier. Right? It can pick up electrons along with hydrogens and move the energy to the next level. So it's a mobile electron carrier. It moves between complexes in the membrane. So the photosystem 2, and then later on the cytochrome B6F. It reduces plastoquinone to plastohydroquinone. Remember that I said essentially when you look and you see the addition of a hydrogen, that's a reduction reaction. So it reduces plastoquinone to plastohydroquinone. And in that process, it pumps hydrogen from the outside to the inside of the thylakoids. So that is the pumping mechanism. All right, so we add the two electrons. They come from the photosystem 2. Okay, so the next step is the this Q cycle or the linear pathway. So there's two ways how the electrons can now go. So you see here photosystem 2, you see the plastohydroquinone, and you see that electrons can now flow either in this linear pathway to plastocyanine in the end and then to photosystem 1, or they can go a different pathway, and that is the Q cycle. That is this cyclic pathway where they pick up hydrogen from the outside and right here, and then can be reused in the next round. And eventually these electrons, they can either continue to flow cyclically, pumping more hydrogen, or eventually they can go through the plastocyanin and then reduce NADP plus to NADPH. So here's the linear pathway, here's the Q cycle, and electrons that come from the plastohydroquinone can either be going this pathway or that pathway, right? And so these electrons that originally come, of course, from the water and then were given up to the photosystem 2, they can be either shuttled this way, or they can be recycled and pump additional hydrogen. So here, this is the recycling, the pumping of additional hydrogen that I highlighted here with green. So that is the Q cycle. So here's the summary formula for electrons as split from the 2pQH2. Two of them reduce the iron-sulfur cluster here, that's in the linear pathway, or they reduce plastoquinone to plastohydroquinone in the Q cycle. That pumps additional hydrogen. Okay. Here is just for you another way to see where there's a possible entry point for electrons from the photosystem 1 from what's called the cyclic electron flow, not the Q cycle. So I'm just pointing this out here for later, where electrons come over here from the photosystem 1 and can be shuttled into this pathway and be added into this complex again. So that's the entry point. All right. Now let's move on to the photosystem 1. So in the end, finally, photosystem 1 reduces NADP plus to NADPH. And so the electrons were coming from the linear pathway from down here, plastocyanin, and you see that all of these letters here, those are all proteins, right? So I told you it's a big protein complex. These are all different proteins that are clustered together, make up this complex. And so the electrons are given from one part to the next part to the next part to the next part, and eventually they get to the point where they can either cyclically flow back to the cytochrome B6F and then be fed into the Q cycle, where they can pump more hydrogens and then eventually go back into the linear pathway, or they can go straight into the linear pathway, pick up hydrogens, combine with the electron that they have, produce NADPH, and produce the major compound then that is required. And so that is the NADPH that is being made. So in the cyclic electron flow, there's more hydrogen pumping that occurs over here, really in the Q cycle, but it allows that extra pumping by flowing these electrons back into the cytochrome B6F. If you have the cyclic electron flow, then that electron doesn't help you produce any NADPH, right? So there's no NADPH if you go into the cyclic electron flow. So when does that happen? Why would that happen? Why would the plant do that? Well, it doesn't sit there here, the electron, and goes, well, should I turn left or should I turn right? These are all chemical reactions that depend on equilibria, right? So if there's a certain amount of compounds that are present, then it's more likely that a different route is taken than if there are fewer compounds of a particular kind that are present. What I mean with that is that the NADPH, if there's a lot of NADPH around, so there's a lot of reduction equivalents around, if that ratio to ATP is high, then there's really no need here to produce a lot more NADPH, but maybe it is important to get more ATP. Well, where does the ATP come from? For that ATP production, you need more hydrogen. And so how do you get more hydrogen? Well, you get more hydrogen by pumping more hydrogen into the lumen, which can then eventually flow through the ATP synthase and produce ATP. So when the NADPH to ATP ratio is high, you get the linear flow that reduces the NADPH to NADH. If you need more ATP, then you will go into the cyclic flow and pump more of the hydrogen in to produce more ATP. And here's the ATP synthase. So this is where photophosphorylation occurs. Here's the ADP. The hydrogen flows through the system. The protein, this protein complex, is able to use the energy from these redox reactions with the electron that is flowing through them to pump out hydrogen and produce ATP. So the H plus gradient that you get that allows for the system to work occurs because you have water oxidation, right? That was the water splitting at the very beginning that I showed you, where you have two waters that produce an oxygen, 4 protons and 4 electrons, and because of the hydrogen pumping into the lumen. And so every time that you get 4 hydrogens flowing through here, you can get 1 ATP. All right. So that's the gist of the light reaction. Now, let me spend a few minutes at with the question of what happens to a plant when there's too much light. Can there be so much as too much light for a plant? And you'll see that yes, there can be too much light, too much of a good thing. And that process is called photoinhibition. So photoinhibition occurs when too many photoproducts, photons, light from the sun, strike the plant and elevate these electrons from a lower level to a higher level. Remember that at that point, these electrons have to go somewhere, right? They can either fall back to a lower state via production of heat or production of light, or they can go into chemical energy. But if there's so many electrons that are coming around, that the plant cannot basically absorb them all at the same time, then what can happen is that instead of producing heat or fluorescence. These photons don't produce photosynthesis, and they don't produce heat or fluorescence. They combine with oxygen that is around in the system, and they produce these reactive oxygen species. So a reactive oxygen species could be O2 minus, or a radical, so this would be O2 plus a single oxygen. Those are oxygen, reactive oxygen species or radicals. They oxidize any kind of protein that they can come in contact with. They're so energetic that they can make these oxidations in a blink of an eye. And of course, that is very bad, right? So if you produce a ton of, if you absorb a ton of photons and you produce a bunch of electrons and you cannot put them into chemical energy or physical energy, then what happens in the plant is that these electrons will find their way to oxygen. They will create these reactive oxygen species, and that is bad. But the plant has a plan in order to prevent this. And so prevention can take a number of forms, two of which I will mention to you here. One of them is to oxidize or destroy the D1 portion. That's just a protein that's part of this large photosynthetic complex PS2. So destroy the D1 part of the protein. And so at that point, that means the photosystem is no longer accepting electrons. So these electrons can't be propelled to a higher level, and they can't then have to find their way either into photosynthesis, falling back down as heat or fluorescence, or produce these reactive oxygen species, because there's no acceptor anymore. The acceptor is destroyed. The plants basically push the self-destruction button. And that seems to be better for them than to deal with potentially having to repair all these different proteins that reactive oxygen species can destroy, when the only protein that needs to be replaced is the D1 protein that the plant destroys itself. How does it destroy that? Well, there isn't really a button that it can push, right? So this is all, chemically, of course, sensed. And the way that that works is that the D1 protein is so sensitive to oxygen, that is to reactive oxygen, that it's oxidized first. It's just so sensitive that that's the first thing that that breaks. And it's also the first, the first set of proteins that electrons will see, if you will, because that is where the light strikes at the photosystem 2, where electrons are evolved, where they are produced from the lower to the higher level of energy, and where they can then immediately be used by this photosystem to destroy the D1 part of the complex if too many of these electrons are around at a time. So oxidation of the photosystem 2, that lowers the photon uptake. So that's one way how plants can deal with photoinhibition. So photoinhibition 1, D1 oxidation, right? That is one pathway. Second pathway of how to deal with too much light, and that's called the xanthophyll pathway. Xanthophylls, xanthos in Greek means yellow, and phyll, of course, is the leaf. So this is not the chlorophyll, not the green part of the leaf, but the yellow part of the leaf. So we have the xanthophyll pathway, and the xanthophyll pathway is comprised of these three types of xanthophylls. There's viola-xanthin, anthera-xanthin, and zeaxanthin. And you see that these products, these compounds have very long carbon-hydrogen chains, and then they have these two ring structures, and both of them contain an oxygen here and an oxygen there. And these substances, the viola-xanthin, can be converted into anthera-xanthin, into zeaxanthin, and it can also go back the other direction. So it can go back from zeaxanthin to viola-xanthin. And in this process of conversion from one to another, that is essentially where electrons are absorbed that are about to create mischief in the plant. And when the high irradiance or the lots of light, the sunlight, when that passes, then viola-xanthin is restored in a reverse reaction. So how does that work? Let's look at this one point at a time, one step at a time. I put these three steps here so you can see where we start. We start with low light, and then we have the second step here. So second step here and here, where we have the addition of these hydrogens, and where that oxygen, together with these two protons from the surroundings, with the help of ascorbate, is made into water. So we're plucking away these oxygens. And ascorbate is a compound that is required as a helper compound during that reaction. And what is ascorbate? Ascorbate is vitamin C, so ascorbic acid or vitamin C. So vitamin C, is a quencher of reactive oxygen species. So plants can use, while they produce vitamin C, they can also use it for their own protection. So it's not just something that animals use as a vitamin. So here's one oxygen that is being picked off and added to hydrogens to produce water, and here's the second one that is being picked off in the second reaction. And so in the end, you get the zeaxanthin, and the oxygens are gone in the form of water together with two hydrogens and the electrons, right? And the electrons, of course, are the problem here. And then if we want to go back and we want to restore this for the next time that zeaxanthin is needed, then oxygen can be used, can be added back, NADPH is required, and you have this two-step process, and the oxygen is added back here and added back there. So let me sum this up here for you so that you can see that for yourself. If you want to stop the lecture here and just sort of read through these things, where do the oxygens come from? Where do the electrons go? What is scavenged? What is added? Where the oxygen's moving from here to the water and in the end back, right? So you have the steps 2 and the steps 3 in the restoration of the zeaxanthin. So that is the photoinhibition pathway, the xanthophyll pathway that allows plants to deal with extra electrons from too much light. And I just want to show you this figure that has xanthophylls on the y-axis. So this is the millimole amount of these xanthophylls per mole chlorophyll. So given the amount, this is basically a ratio of xanthophylls to chlorophylls. Here's a time axis during the day, and then here's the amount of light that occurs. So here's sunrise, and we have no light, and then the light becomes more and more intense. In bright daylight, we have about 2,000 micro-Einsteins or micromoles per square meter per second. So this is a very sunny day here. It's about 2,000. And then at 6 o'clock, this sun sets here in this particular experiment. But the point that I'm trying to make is that we're starting with a pool of zeaxanthin that can be used in photoinhibition. And you see how it dips during the day. And at the same time, these compounds, zeaxanthin and anthraxanthin, are increasing in their concentration, and as they are then when the highest amount of light of the day is over, they can be reverted back to zeaxanthin. So it reduces the number of these compounds is reduced, zeaxanthin is increased, and so we have a condition back at the beginning for the next day when light may become a problem again. So that is the end of the lecture for the light reaction and the photoinhibition.